Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12th Molecular Basis of Inheritance, that is part 13. Regulation of gene expression. Regulation of gene expression refers to as in a broad term that maybe occurs in various levels. Means the regulation takes place as in the different levels. They are considering as in gene expression result in the formation of the polypeptide. It can be regulated as in several levels. When you observe to as in the eukaryotic cells, the total regulation takes place as in different levels. Okay, let's give in here the eukaryotes in the regulation. They are extended by 8. First one is the transcriptional level, means the formation of the transcript. So the transcription will be that mostly the regulation takes place in where that in a different levels, right? So that regulation where they complete the control either in a first the transcription level or in the processing level, means that is the regulation of the splicing process, means the formation of the HNRNA, not in a HNRNA in the splicing process also you get complete the regulates, and then the transport of the mRNA from the nucleus to as in cytoplasm. So for example here has it that enter the nucleus is present. So in this nucleus to this is a complete what is a part of the cytoplasm, right? So nucleus to the cytoplasm here also to get regulates, right? And next that is a translation level, that is in a translational level. So in these different levels we completely to get their regulates as in a the gene. Okay, let's after the gene expressed to perform as a particular function or a set of the functions. So each and every the gene as in the performed as in a only in a common function, as in a the common function or a set of functions, or a set of the functions the gene to perform as in a function. Here for example the given the enzyme is called as a beta galactose. The given as one enzyme is beta galactose is in the synthesis of Escherichia coli. It is catalyst as in hydrolysis of disaccharides. It is converted into the mostly the disaccharide molecule. What is the disaccharide molecule? That is a lactose. Here is in the lactose molecule. It is a disaccharide molecule. In this disaccharide the completely the lactose into convert as what in a glucose and galactose. In this disaccharide molecule, it is completely the hydrolysis process. It is breakdown to form as what? One is in a that is galactose. One is for as in the galactose, and the one is in what? To formation of in a the glucose also. The formation of what? That is glucose also. Okay, and the bacteria use them their source of the energy. So when the glucose galactose is a simple molecules, am I right? So in this glucose and galactose the bacteria, means that Escherichia coli, to get utilize the energy source to that would be no longer required as a synthesis of the enzyme that is in a beta galactose. So it is no requirement is there for the energy synthesis. Okay, therefore the simple terms, it is the metabolic the physiological environmental conditions is regulated by as in the expression of gene. So the when the environment factors will be there completely differ, then what happen? The time only to get automatically the genes will be there completely to get that depends upon the beta galactose. Okay, like the development and the differentiation of the embryo into the adult organisms also result in a coordinate regulation the expression of the several set of the genes. So here the adult organisms to get the, before we called as say before we use one word, that is what the gene perform as in the common function or a set of functions. So it is adult organisms here mostly the expressed as in the set of the genes. Here the set of the genes, and then here once you observe as the prokaryotic cells, here the prokaryotics, the prokaryotes control the rate of the transcriptional initiation is the predominant set control of the gene expression. So here mostly the prokaryotic cells, the mostly the transcription takes place only where in the cytoplasm only. They are not, for example, when you take as in any one particular cell, it is in the cell, it is in a nucleus, it is in a cytoplasm. Am I right? So here only the transcription takes place. Here also only translation takes place in the prokaryotic cells. So in the transcriptional unit, the activity of the mRNA, the mostly polymerase, the given as a promoter is as in the term regulated by interaction with as an necessary proteins. These which are affected, it is ability to recognize and start the sites. So when their requirement is there, then only what happens, the set of the enzymes they get participate in the regulation, they start with as in their function. Okay, like these are regulatory proteins can act as in both positively means activator and then negatively the receptors. So they act as in mostly in activators as well as in they act as in repressors means activated and deactivated like. Okay, next the accessibility of the promoter, the region of the prokaryotic DNA is the mainly causes the regulated by as in the interaction of the proteins with as in sequences, the term is called as operators. So which one they get acting as in the major function. So those are called as what? Promoters. We call as what? Promoters. Am I right? So here that promoters to get completely they get start their function, their mediators they require which purpose, they performance they complete they get operated, right? So those are called as an operators. Those are called as what? Operators. That operators to get completely regulates as in this function. Okay, this operator regulation, the adjustment of the promoter, the elements must be the operons, and the most of the cases in the sequence of the operator bind with as in a repressor protein. So mostly when the repressor protein here, for example, when we take as in the operator region, when we see as in the operator region, they're complete to get bind with as in the repressor protein, to get bind with as a repressor protein. Then what happened? They perform as in different functions. Each operand it has in a specific operator, the specific repressor. So each one has in a what? In specific complete repressor is present, specific operator also is present. For example, they given here the lac operator is the presence of only for the lac operand only, and then it is in interacts with a specific with as in a lac repressor only. So here the lac operand they are participate as an only for the lac only, lac operand initiate as any only for as any lac of the repressors. Okay now let's once we observe briefly in that in a lac operand concept. Let's once we observe lac operand concept. See the first one, the lac operand. The lac operand. Here, lac means what? The lac or nothing but as in a glucose. Am I right? That is in a glucose molecule. Operand means that is a concept. Here, the glucose molecule, it is there completely partially to get divided and to get produced as in a what? The sugar molecules, like as in glucose and galactose. Am I right? Let's see. Here, the elucidation of lac operand was first the result the closely association between the most genetist, the Francois Jacob and biochemist that is named the Jacques Monod. In this both scientists that first they explain as their lac operand concept. The scientist was in a Jacob and Monod. In this scientist, one was in a genetics, one was in a biochemist. In this scientists, their first they are explained about as in a what? Lac operand concept. They were the first elucidated as in the transcriptionally regulated by the system. In the scientists, they explain about as in a that how do the total transcription will be the regulated. They did a first experiment. In the lac operand, you can see here lac means it's a lactose molecules. But it's a part of the disaccharide molecule. And polycistrons, the structural genes, the regulated by as in the common promoter regulated genes. So when you observe to as in these polycistronic genes in that lactose molecule, this lactose, the lac means their lactose molecule. In this lactose, it is converted as what? In a glucose as well as in a galactose. Am I right? Galactose. Let's in this such arrangements as very common in the bacteria, it refers to as an operand. It is this total mechanism is common in that in a bacterial cells. So in this common, it is referred to as what? Operand. The name few such example they given here, the lac operand and tryptophan and era operand, his operand and valine operand, etc. Like the different types of their operands as present. Okay, and then the lac operand consists of as the lac operand consists of one of the regulatory gene that is in I gene. The regulatory gene is present, that is what? I gene. And here the in the term I does not refer to the end user. It is not differ to as any inducer. Rather, it is divided from as an inhibitor. So here I gene, this their I gene, it is mostly regulated by as an inducer only. Okay, there are three structural genes as present, that is Z, Y, A. Here has in A, there are three regulated genes as present, that is in A, Z, Y, A. Their I gene codes for as in that repressor, the lac operon. Y gene codes for as an beta galactose, and which is primarily they're completely responsible for as a hydrolysis of the disaccharides. Lactose, it is that monomeric units like as in glucose and galactose. See, once I'll explain this diagram, see here they given clearly. See here, when the here mostly the glucose requirement is present, here then what happened? They participate in completely the galactose enzyme. But see here, mostly in this Y, let's I'll tell you. The Y gene code for as in the permease, which is in the increase the permeability as in the beta galactose. The A gene codes for as in the transstylase. Hence, these are three genes are products. Lac operon is required for as in metabolism of lactose. So when the mostly the bacteria is requirement as in that energy in these three means A, Z, Y, A genes are there mostly important for as in a to get the synthesis of any molecules. The most other operators as well as in a genes present as in the operon are needed to gather to function of the same regulated metabolic pathway. So in this metabolic pathway, this all is very, very requirement. Let's see once I explain this diagram. See in this lac operon. So here they are completely in this first one P is in a promoter sites. O for as an operator. O for as what? Operator. I means I gene. Here ZYA. In these three are called as a structural genes. ZYA. Here absence of inducer. Here absence of it. For example, here we observe as an one culture plate. Here has in what bacteria is present. Escherichia coli. If in case here completely you keep here some glucose molecules and then galactose molecules. These are the glucose, and then these are the galactose, and then these are the lactose molecule. These are the lactose molecule. So in this medium, here complete the glucose is present. Glucose is present as well as in A, their galactose is present. Galactose is present. Here no requirement of in A, their inducer, because here only glucose is present and galactose is present. So the bacteria to get easily to get utilized as in A, here completely glucose and galactose and they survive easily. And means here complete their absence of inducer, means here complete is a part of what? It is completely in a switch off. It is in what? In a, they are part of in a their switch off. It is in a switch off. Let's see here. In this process, the repressor, they get bind with as an operator. It is completely here repressor to bind with as an operator region. The prevents as an RNA polymerase from as a transcription in the operon. Let's see here what happened. So in this process here completely, mostly this all thing is complete. Only has in what? In a the lactose molecule only is present. Here has in only for lactose molecule. So here inducer is present. So when the inducer is present means energy requirement is present. Then what happened here? The mostly here in this inducer to get inactivated. In this mostly what happened in this process? The repressor mRNA, it is to get activated with help of as an RNA polymerase. Then it is activated. Then what happened in this structural genes ZYA? See in this ZYA, in this beta galactose, it is to get completed synthesis as an they are disaccharide molecules. It is they get hydrolysis, simply called as an they are hydrolysis. It is to get formed completely to break down to as in a the simple molecules, and permease to give as a permission to as it get here to here. In this trans-acetylase, it is the trans-acetylase, it is to get help to as in to get synthesis of in a protease, means in this process of in a the translation process they are help to as in a gene. Okay, now this is the in a part of the diagram in a lac operon. See, they given here clearly. Mostly when you observe, the lactose is the substrate for as the enzyme beta galactose, and it is the regulate switch, complete the switching on and off the operon. So before when you using the switch off and switch on, right? Hence the term as an inducer. It is very important, inducer. So inducer, they get act as a switch off and switch on. Their absence of the in the presence of the carbon source such as in a glucose and lactose is provided as in a growth of the medium, the bacteria. So in this growth of medium, here the glucose is present. What happened? The galactose is given to as in a that energy source, right? The lactose is transported into the cells through the action the permease. So the given is what? Permease function is to give as in permission, means to give as in that action, right? Here the remember very low level of the expression of the lac operon has to be the present in the cell all the time, otherwise the lactose cannot be entered into the cell. So then what happened here? The permease is not there. What happened? It is not to give as a permission, am I right? Like here lactose then include the as an operate the operon, it is in a following manner. So when the lactose molecules, it is there followed as in a, there completely there several manners. They are given as in a first one, the repressor of the lac operon. The operon synthesized all time continuously, so constantly from as an I gene. So I gene, regulated gene is there now. The I gene to get continuously they get synthesis as takes place. Then repressor protein binds with as an operator region of the operator, the prevents as an RNA polymerase from the transcription operon. So here what happened? They are bind with as a that in a repressor into as an RNA polymerase. Then in the presence of the inducer such as in a lactose or allolactose, it is a repressor the inactivated by as in a the interaction with as an inducer. So your lactose molecule they go to as what? It go to as an inactivated condition. After this interaction is takes place as what? The automatically the inducer will be they get stopped. This allow RNA polymerase the axis, the promoter and transcription process, the produce the essentially the regulation of the lac operon can be also visualized the regulation of enzyme synthesis. It's in a substrate. So then finally what happened? The gene regulation takes place. Then what happened? To synthesis takes place in the process of in a they are the translation process by as in a synthesis of in a substrate. Okay, now this is. Remember the glucose galactose cannot act as an inducers for lac operon. Can you think how long lac operon would be the expressed the presence of the lactose? Because they are using as in the hydrolysis process, it is converted as what? In a glucose as well as in a galactose. Okay, now next the regulation of the lac operon by the repressor is referred to as in the negative regulation. The lac operon under control the positive regulation, well, but very behind the scope of discussion at the level. So the lac operon, the repressor is always there negative, but under control the some enzymes, any the regulation will be takes place. What happened? It is converted as in a positively as well as behind scope of the discussion about as in this total process. Okay, now this is incompletely what in a lac operon concept. Okay, remaining we discuss in next class. Thank you for watching. Please like, share and subscribe.